Life on Earth began a very long time ago. The Cambrian Period was a truly special time in our planet's history. Many new and diverse types of animals appeared quite suddenly in the oceans. Scientists call this remarkable event the Cambrian Explosion. This burst of life happened roughly 540 million years in the past. Our own evolutionary story, the story of vertebrates, starts in these ancient, teeming seas. We must search for the very first indications of our own distinct group. Vertebrates are defined as animals that possess a backbone. Understanding their ancient origins is a fascinating scientific endeavor. These initial evolutionary steps were incredibly important for all that followed. Before the development of true backbones, there were creatures known as chordates. These early animals shared several key anatomical features. They all possessed a notochord. The notochord was a flexible, rod-like structure that provided bodily support. Another shared characteristic was a dorsal hollow nerve cord. This structure would eventually evolve into the spinal cord and brain in later vertebrates. Pharyngeal slits were also present, located near the throat region. These slits were likely used for filter feeding or for respiration in these aquatic organisms. Finally, a muscular post-anal tail extended beyond the anus, aiding in propulsion and swimming. These fundamental features collectively define the phylum Chordata, our broader animal family. The very first true vertebrates to appear in the fossil record were the jawless fish. Paleontologists refer to this group as the agnathans. These pioneering fish emerged in the ancient Cambrian seas, marking a significant evolutionary milestone. Well-known examples from fossil discoveries include creatures like these early vertebrates already had a distinct head region and developing sensory organs. They possessed a cranium, a protective structure made of cartilage or bone, enclosing a simple brain. Numerous gill slits were present along their sides, used efficiently for extracting oxygen from the water. Crucially, however, these early fish entirely lacked jaws. They also did not possess the paired fins that are characteristic of most later fish species. These agnathans were true pioneers, showcasing the earliest vertebrate body plan. These primitive jawless fish led a relatively simple existence in the ancient oceans. Many of them were probably filter feeders, patiently sifting small food particles from the muddy seabed. Alternatively, they might have actively sucked tiny organisms and organic detritus directly from the water column. Their bodies were often covered in an armor of small bony plates or scales. This external plating would have offered some degree of protection from any predators that existed at the time. However, their lack of jaws severely limited their dietary options and feeding strategies. Their swimming capabilities were also likely not very powerful or agile compared to later fish. Despite these limitations, they represent a crucial and foundational early stage in our deep vertebrate lineage. The evolutionary development of jaws represented a truly massive and transformative change for vertebrates. This significant biological innovation occurred primarily during the Silurian period. This geological period commenced approximately 440 million years ago. The advent of jaws opened up an entirely new world of ecological possibilities for fish. With jaws, fish could now effectively bite, grasp, and process prey. This ability turned them into much more formidable and efficient hunters. The presence of jawed predators dramatically altered the dynamics of life within the ancient oceans. The evolution of jaws was undeniably a key and pivotal step towards the more complex vertebrate forms that followed. Jaws did not simply appear out of thin air as entirely new structures. Current scientific understanding suggests that they evolved from pre-existing structures, the gill arches. Gill arches are the series of bony or cartilaginous supports that hold the gills in place. The very frontmost pair of these gill arches gradually underwent modification. Over evolutionary time, they became larger, stronger, and crucially hinged. This transformation resulted in the formation of the first simple yet functional jaws. True teeth, which are distinct structures, evolved somewhat later, making these newly formed jaws even more effective tools for feeding. This modification was a brilliant and elegant example of natural selection at work. Evolution effectively repurposed an existing anatomical structure for an entirely new and advantageous function. Early jawed fish included several fascinating and diverse groups, such as the plecoderms. Placoderms were a group of often heavily armored prehistoric fish. The formidable was a giant apex predator among the placoderms with powerful blade-like jaw plates. Another significant early group was the acanthodians. These fish are sometimes colloquially referred to as spiny sharks, though they are not directly related to modern sharks. They were characterized by having many small, sharp spines supporting their fins. These early jawed fish were remarkably diverse and ecologically successful during their time. They clearly demonstrated the profound advantages conferred by possessing a biting apparatus. They could now exploit a much wider range of food resources than their jawless predecessors. The appearance of jaws had an immense and far-reaching impact on the structure of marine life. Jawed fish rapidly rose to prominence, becoming the dominant predators in many aquatic ecosystems. This, in turn, triggered what is known as an evolutionary arms race. Prey animals were under intense pressure to evolve better defenses, such as thicker armor or camouflage. Other fish species evolved to become faster and more agile swimmers to escape predation. The entire marine ecosystem became significantly more dynamic and complex as a result. The presence of jaws was a powerful driver of diversification among fish. It effectively set the stage for the subsequent evolution of all jawed vertebrates, a lineage that ultimately includes ourselves. This was truly a revolutionary development in the history of life. The Devonian period is very often referred to by paleontologists as the Age of Fishes. This fascinating geological period began approximately 419 million years ago. It was a time that witnessed an extraordinary diversification and flourishing of fish species in the world's oceans and freshwater systems. During the Devonian, the bony fish, scientifically known as Osteichthyes, became particularly common and widespread. These fish possess skeletons primarily composed of hard bone, rather than the softer cartilage found in sharks. Many bony fish also had a gas-filled sac called a swim bladder, which helped them control their buoyancy in the water. This era was a crucible of major aquatic evolution, with many novel fish forms appearing in the ancient waters. The diverse group of bony fish ultimately split into two principal evolutionary lines. These two major groups are the ray-finned fish and the lobe-finned fish. The ray-finned fish are scientifically classified as Actinopterygii. Their fins are characteristically supported by numerous thin, bony spines or rays, much like the struts of a fan. Today, ray-finned fish represent the most diverse and species-rich group of all vertebrates on Earth. Indeed, most of the fish species that you are familiar with, from goldfish to tuna, are ray-finned fish. The lobe-finned fish, on the other hand, are known as Sarcopterygii. Their fins are structurally quite different. They possess a more fleshy, muscular and robust fin structure, which proved to be incredibly important. Lobe-finned fish are distinguished by having fins that contain a central, bony skeleton. These internal bones are remarkably homologous to the limb bones found in terrestrial vertebrates, including our own arms and legs. This homology means that they share a common evolutionary origin and underlying structural plan. Robust muscles extend down into these fleshy fins from the body. This muscular arrangement gives their fins considerably more power and a greater range of flexible movement compared to ray fins. Living examples of lobe-finned fish include the famous coelacanths, once thought long extinct, and the various species of lungfish. Lungfish possess the remarkable ability to breathe atmospheric air using lungs in addition to their gills. These specific features were profoundly important for the evolutionary events that were to follow. They held the anatomical key to life on land. The lobe-finned fish were absolutely crucial for the momentous transition of vertebrate life from water to land. Their sturdy, muscular and fleshy fins were, in a sense, pre-adapted for a new role. These fins could be effectively used for support in very shallow water environments. They could help a fish to prop its body up off the substrate, perhaps to navigate through dense aquatic vegetation, or to reach the water's surface to gulp air. This was a significant behavioral and anatomical step towards true walking on land. The arrangement of bones inside these ancient lobe fins is strikingly similar to the pattern of bones found in our own arm and leg limbs. Therefore, these ancient fish carried within their bodies the fundamental skeletal blueprint for all future tetrapod limbs. Our own limbs, essential for our way of life, began their evolutionary journey with these remarkable fins. Making the evolutionary move from a fully aquatic existence to life on land was an immense and formidable challenge for early vertebrates. The terrestrial environment presents a drastically different set of physical conditions compared to water. Air is far less dense and supportive than water, therefore, animals venturing onto land need strong limbs and skeletons to support their body weight against the pull of gravity. Air also causes things to dry out very quickly through evaporation. Consequently, terrestrial animals needed to evolve effective ways to prevent dehydration and conserve body water. Furthermore, gills, which are highly efficient for extracting oxygen from water, do not function well in air. Lungs or similar air-breathing structures became absolutely essential for respiration on land. These were major physiological and anatomical hurdles that aquatic life had to overcome. A truly remarkable fossil discovery, a creature named, provides invaluable insights into this critical water-to-land transition. Lived approximately 375 million years ago, during the late Devonian period. It is considered by scientists to be a perfect example of a transitional fossil, beautifully illustrating an intermediate stage. Possessed a fascinating mosaic of features, some fish-like and some tetrapod-like. Its fish-like characteristics included scales covering its body, fins for propulsion in water, and likely functional gills. However, it also exhibited several features typical of early four-limbed vertebrates, tetrapods. It had a flattened head with its eyes positioned on top rather than on the sides like most fish. It also possessed a mobile neck, allowing it to turn its head independently of its body, a feature absent in most fish. Crucially, its pectoral fins contained wrist bones and finger-like bones, robust enough to support its body in shallow water or on land for short periods. Following creatures like the first true tetrapods or four-limbed vertebrates made their appearance in the fossil record. These were the early amphibians. Well-studied examples of these pioneers include, and they lived towards the end of the Devonian period, representing the next step in terrestrial adaptation. These animals had clearly defined limbs with distinct digits, fingers and toes. They were capable of walking on land, although their locomotion was likely somewhat clumsy and sprawling compared to later terrestrial vertebrates. However, despite these advancements, these early amphibians were still very closely tied to aquatic environments. Their skin was not waterproof and was prone to drying out. They also crucially needed water for reproduction, as their eggs lacked a protective shell. Early amphibians evolved several important adaptations that suited their semi-aquatic lifestyle. They developed stronger and more robust skeletons to adequately support their bodies against the constant pull of gravity on land. Lungs became the primary organs for breathing atmospheric air, although some species may have also retained gills or used their moist skin for gas exchange to some extent. Their newly evolved limbs allowed them to move around on land, perhaps to find new food sources like insects, or to escape from aquatic predators. However, a significant constraint remained. They typically laid their soft, jelly-like eggs in water. The larval stage of amphibians, like the familiar tadpoles of modern frogs, was entirely aquatic, breathing through gills. This characteristic dual life, partly on land and partly in water, continues to define amphibians today. Section 5. Conquering the Land, the Reptilian Ascent. Amphibians certainly made the first courageous and significant steps onto the terrestrial landscape. Despite their pioneering efforts, they remained fundamentally tethered to aquatic or very moist environments. Their permeable skin, which often assisted in respiration, needed to stay consistently moist to function properly and to prevent lethal dehydration. Furthermore, their eggs were soft and lacked any substantial protection against drying out if laid on land. This biological constraint severely limited their ability to explore and colonize drier, more arid inland habitats. A new group of vertebrates, however, evolved features that allowed them to overcome these critical amphibian limitations. These animals were the reptiles. They became the first vertebrates to truly conquer the terrestrial world, adapting in remarkable ways for life away from water. The single most significant and revolutionary reptilian innovation was the development of the amniotic egg. This was a profound evolutionary breakthrough. The amniotic egg possesses a protective, often leathery or hard, outer shell. This shell provides physical protection and, crucially, prevents the delicate embryo inside from drying out, even in arid conditions. Internally, the amniotic egg contains several specialized membranes that support the developing embryo. The amnion is a fluid-filled sac that encloses the embryo, creating a kind of private pond and cushioning it. The allantois serves as a storage sac for metabolic wastes and also plays a role in gas exchange. The chorion, another membrane, facilitates gas exchange between the embryo and the outside environment. Finally, the yolk sac contains a rich supply of nutrients to nourish the growing embryo. This remarkable structure allowed reptiles to reproduce successfully on dry land, independent of water bodies. In addition to the amniotic egg, reptiles evolved a suite of other key anatomical and physiological features that enhanced their survival on land. They developed tough, dry, scaly skin. This skin is largely waterproof, forming an effective barrier that helps to prevent excessive water loss from the body through evaporation. Their lungs became more efficient and complex than the simpler sac-like lungs of most amphibians. Reptiles typically use ribcage expansion and contraction to draw air into their lungs, a more powerful mechanism than the buccal pumping seen in amphibians. Reptiles also generally possess stronger limbs. In many reptilian groups, their limbs were positioned more directly underneath the body, rather than sprawling out to the sides. This more upright posture allowed for more efficient and often faster movement on land. These combined adaptations made reptiles exceptionally well suited for life in drier, fully terrestrial climates. With these advantageous adaptations firmly in place, reptiles thrived and underwent a massive diversification. They spread rapidly across the globe during the Permian and most notably the Mesozoic eras. In fact, the Mesozoic era is often famously referred to as the Age of Reptiles due to their dominance. Dinosaurs, perhaps the most iconic group of extinct reptiles, roamed the continents in a vast array of forms. Flying reptiles called pterosaurs soared through the skies. Meanwhile, various groups of marine reptiles, such as ichthyosaurs and plesiosaurs, re-inhabited the oceans, becoming formidable aquatic predators. Early ancestral reptiles included forms like a small lizard-like creature from the Carboniferous period. This great evolutionary radiation of reptiles clearly demonstrated the profound success of the amniotic egg. It also highlighted the effectiveness of their other adaptations for a fully terrestrial existence. Section 6, The Dawn of Mammals, from Synapsids to Furry Critters. While the great dinosaurs and other reptiles ruled the terrestrial ecosystems during the Mesozoic era, another distinct group of amniotes was quietly evolving in their shadow. This group was the Synapsids. Synapsids represent one of the major branches of the amniote evolutionary tree. They are distinguished from other amniote groups, such as sauropsids, which include reptiles and birds, by a specific feature in their skull anatomy. Synapsids possess a single opening, or fenestra, in the skull located behind each eye socket. This opening is termed the temporal fenestra. This anatomical feature is thought to have allowed for the attachment of larger, more powerful jaw muscles, improving biting force and feeding efficiency. Modern-day mammals are, in fact, the sole surviving members of the synapsid lineage. Our own evolutionary heritage traces directly back to these early synapsid forms. Their story is one of gradual, step-by-step transformation over many millions of years. Over extensive periods of evolutionary time, certain lineages within the synapsid group became progressively more mammal-like in their anatomy and physiology. These transitional forms were often historically referred to as mammal-like reptiles, although this term is somewhat misleading as they were not true reptiles. A more accurate scientific description is stem mammals or non-mammalian synapsids. Early examples of synapsids include creatures such as Dimetrodon, which is famous for the large, sail-like structure on its back, though it was a very early form. Later, a more advanced group of synapsids, called the therapsids, evolved. This diverse group included the cynodonts. The cynodonts, in particular, exhibited many features that were remarkably close to those seen in true mammals. For instance, they had differentiated teeth, incisors, canines and cheek teeth, adapted for more efficient food processing. Their posture was also becoming more upright, with limbs positioned more directly beneath the body. They were clearly becoming increasingly active and metabolically sophisticated creatures. The complete evolutionary transition from these advanced cynodonts to the first true mammals involved the acquisition of several key defining characteristics. Mammals are endothermic, meaning they are warm-blooded. They generate their own internal body heat, allowing them to maintain a constant, relatively high body temperature irrespective of the ambient environment. Fur or hair evolved as an insulating covering to help retain this body heat. Mammary glands, which produce nutritious milk for feeding their young, developed and became a defining feature of all mammals. Indeed, the very name mammal refers to these glands. Our teeth became highly specialized into distinct types: incisors for nipping, canines for piercing, and premolars and molars for grinding and shearing food. Furthermore, mammals possess 3 tiny bones in their middle ear, the malleus, incus, and stapes, that transmit sound vibrations. These ear ossicles ingeniously evolved from bones that were part of the jaw joint in their synapsid ancestors. The first animals that can be classified as true mammals made their appearance in the fossil record during the late Triassic period. This significant evolutionary event occurred approximately 225 million years ago. These earliest mammals lived throughout the entirety of the Mesozoic era, an immense span of time often dubbed the Age of Reptiles. During this long period, these early mammals were mostly quite small in body size, often comparable to shrews or mice today. Many of them are thought to have been nocturnal, meaning they were primarily active during the night. This nocturnal lifestyle may have helped them to avoid predation by the much larger and more numerous dinosaurs that dominated the daytime world. Their diet likely consisted mainly of insects, worms, and perhaps small vertebrates. Despite their diminutive size and subordinate ecological roles, these early mammals were highly successful survivors. They quietly laid the crucial evolutionary groundwork for the spectacular radiation of mammalian life that occurred after the dinosaurs vanished from the Earth. Section 7, A New Beginning, the Rise of Primates. The Mesozoic era, the long reign of the dinosaurs, came to a sudden and dramatic end. Approximately 66 million years ago, a very large asteroid or comet collided with Earth near the Yucatan Peninsula in modern-day Mexico. This cataclysmic impact triggered the KPG extinction event, formerly known as the KT extinction. This event led to the extinction of the non-avian dinosaurs, along with many other species of plants and animals across the globe. This catastrophic occurrence profoundly reshaped the biosphere. It created numerous empty ecological niches as dominant groups like the dinosaurs were removed from ecosystems. These newly available openings provided significant evolutionary opportunities for the groups that managed to survive the extinction. Mammals, having persevered through the Mesozoic, were among these fortunate survivors. They began to diversify rapidly, expanding into these vacant roles in this new post-dinosaur world. Among the diverse mammalian groups that flourished in the wake of the KPG extinction were the early primates. The first fossil evidence of primates, or their close relatives, appears in the Paleocene and Eocene epochs. These geological epochs immediately followed the extinction of the dinosaurs. These earliest primates were generally small-bodied creatures. They were most likely arboreal, meaning they were adapted to a life spent primarily in trees. Fossil forms such as Plesiodapis from North America and Europe are sometimes considered to be very early primate relatives, or stem primates, although their exact placement is debated by scientists. Other fossil groups like the Adapiforms and the Homomorphiforms are more clearly recognized as true early primates. These creatures adapted skillfully to a life of climbing, leaping, and navigating through the complex three-dimensional environment of forest canopies. Primates, as a group, evolved a distinctive suite of anatomical and behavioral features. These characteristic adaptations were crucial for their success and survival in arboreal habitats. They developed grasping hands and feet, a hallmark of the primate order. These hands and feet often featured opposable thumbs or opposable big toes. This opposability allowed them to grip onto branches securely and manipulate objects with considerable dexterity. Their eyes gradually shifted to a more forward-facing position on the skull. This resulted in stereoscopic or three-dimensional vision. Stereoscopic vision is exceptionally useful for accurately judging distances, a critical skill when leaping between branches high above the ground. Primates also generally have larger brains relative to their overall body size compared to most other mammals. Furthermore, most primate species have flat nails on their fingers and toes instead of the sharp claws found in many other mammals. These early primate traits, developed and refined in the trees, proved to be profoundly significant for subsequent primate evolution. Grasping hands, initially for secure locomotion, also allowed for the detailed manipulation of food items and, much later in our lineage, objects that would become tools. Forward-facing eyes, providing enhanced depth perception, were not only useful for navigating complex forest environments, but also for spotting food or predators. The increased brain capacity, even in early forms, set the stage for the development of greater intelligence, complex social behaviors, and problem-solving abilities seen in later primates. These adaptations, honed meticulously in an arboreal setting, provided the essential foundation upon which all later primate evolution was built. They were fundamental for the subsequent emergence of monkeys, apes, and ultimately our own human lineage. Our most cherished human characteristics have incredibly deep evolutionary roots in these early tree-dwelling ancestors. Section 8. Our closest kin great apes and early hominins. Within the diverse order of primates, evolutionary processes continued to shape various lineages. One particularly significant lineage led to the evolution of the apes, scientifically known as the Hominoidea. The first apes made their appearance in the fossil record during the Oligocene and Miocene epochs, roughly between 34 and 5 million years ago. Apes can be distinguished from monkeys by several key anatomical characteristics. Most notably, apes do not possess an external tail, whereas most monkey species do. Apes generally have broader chests and a different shoulder blade position compared to monkeys. Their shoulder joints are also more mobile and flexible. This enhanced shoulder mobility allows for a wide range of arm movement, including the ability to swing and hang from branches, a form of locomotion known as brachiation. These distinctive features reflect adaptations to different modes of movement and lifestyles within various primate groups. The living great apes are our closest surviving biological relatives in the animal kingdom. This esteemed group includes the orangutans of Asia and the African apes, gorillas, chimpanzees, and bonobos. Humans, Homo sapiens, are also unequivocally part of this great ape family. Extensive genetic studies, particularly the comparison of DNA sequences, have revealed just how closely related we are to these other primate species. Our DNA is most similar to that of chimpanzees and bonobos, indicating a relatively recent shared ancestry. Scientists estimate that we shared a common ancestor with chimpanzees and bonobos until some point in the late Miocene. This common ancestor lived in Africa millions of years ago. Therefore, studying the biology, behavior, and genetics of living apes provides invaluable insights that help us understand our own evolutionary history and unique human characteristics. The specific evolutionary line that eventually led to modern humans diverged from the lineage that led to modern chimpanzees and bonobos. This crucial split in our ancestry is estimated by genetic and fossil evidence to have occurred approximately 6 to 7 million years ago during the late Miocene epoch. The earliest members of our distinct human lineage appearing after this divergence are collectively referred to as hominins. A key and defining characteristic that distinguishes early hominins from other apes is the development of habitual bipedalism. This means consistently walking upright on two legs as the primary mode of locomotion. The adaptation of bipedalism appeared very early in our evolutionary journey. It had profound consequences, most notably freeing the hands from a role in locomotion, allowing them to be used for carrying objects, tool use, and other complex manipulations. This was a crucial initial step on the long road towards becoming human. Several genera of early hominins have been discovered through painstaking paleontological work, primarily in various parts of Africa, which is considered the cradle of humankind. Sahelanthropus tuchadensis, discovered in Chad, is one of the oldest known potential hominins, dating back to around 7 million years ago. Aurorin found in Kenya, lived approximately 6 million years ago and shows some evidence of bipedalism in its femur. Ardipithecus ramidus, affectionately nicknamed Ardi, lived in Ethiopia about 4.4 million years ago and provides a more complete picture of an early hominin. These pioneering early hominins exhibited a fascinating mosaic of anatomical features. Some of their traits were still quite ape-like, such as relatively small brain sizes and adaptations for climbing. Other features, however, were more human-like, particularly the emerging evidence for upright walking. They represent the very beginning of our unique and remarkable evolutionary journey. Section 9. The Human Spark. The Journey of Genus Homo. Our very own genus, Homo, made its evolutionary debut in Africa. Current fossil and archaeological evidence suggests this occurred sometime around 2.5 to 3 million years ago. The appearance of the genus Homo marked a truly significant shift in the trajectory of hominin evolution. Members of this genus, when compared to earlier hominins like Australopithecus, generally exhibited notably larger brains relative to their body size. Alongside this increase in brain capacity, we also see the first clear and consistent evidence of stone tool making and use. This newfound ability to deliberately manufacture and utilize tools was a revolutionary development. It allowed these early humans to access a wider range of food sources, such as scavenging meat from carcasses or processing tough plant materials. It also helped them to process food more efficiently, potentially freeing up energy for further brain development. One of the earliest recognized species within our genus was Homo habilis. Its scientific name translates appropriately to handy man, reflecting its association with early tool use. Homo habilis is linked to the Oldowan tool industry. Oldowan tools were relatively simple, consisting of stone cobbles crudely flaked to create sharp edges for cutting or scraping. Later in time, another important species, Homo erectus, appeared on the evolutionary stage around 1.9 million years ago. Homo erectus possessed an even larger brain than Homo habilis and had a more modern human-like body plan. They are credited with developing more sophisticated stone tools, known as Acheulean tools. These included characteristic teardrop-shaped hand axes, which were versatile implements. Significantly, Homo erectus was the first hominin species known to have migrated out of Africa, spreading into parts of Asia and Europe. They also likely learned to control and use fire, a major technological and cultural advancement. Other fascinating Homo species followed Homo erectus in the hominin family tree. These are often collectively referred to as archaic humans. One of the most well-known of these is Homo neanderthalensis, commonly known as Neanderthals. Neanderthals lived primarily in Europe and parts of Western and Central Asia from about 400,000 to 40,000 years ago. They were remarkably well adapted to the cold climates of Ice Age Eurasia. They had large brains, comparable in size to our own, and a robust muscular build. Neanderthals manufactured complex stone tools, the Mousterian industry, and were skilled hunters of large game animals. There is also evidence that they buried their dead and may have created simple forms of art or personal adornment. The Denisovans are another enigmatic group of archaic humans, known mainly from limited fossil remains and more extensively from ancient DNA extracted from these fossils. Compelling genetic evidence shows that our direct ancestors, early Homo sapiens, interbred with both Neanderthals and Denisovans during their expansion across Eurasia. Anatomically modern humans, our own species, Homo sapiens, evolved in Africa. The earliest fossil evidence for Homo sapiens dates back to around 300,000 years ago. These early members of our species showed clear signs of advanced cognitive abilities and complex behaviors. They developed increasingly sophisticated and diverse toolkits, including blades, projectile points, and tools made from bone and antler. They created remarkable works of art, such as the famous cave paintings found in Europe and other parts of the world. They used symbols extensively and likely developed complex, nuanced language for communication. Starting around 70,000 to 100,000 years ago, populations of Homo sapiens began to expand out of Africa in significant waves. They eventually populated almost every habitable corner of the globe, adapting to a vast range of environments. Our incredible journey from ancient jawless fish in primeval seas to modern humans capable of contemplating our own existence is truly an epic tale. We are, in every sense, a remarkable product of billions of years of evolutionary processes.